to use in the entire module. Screen is visible? Yeah. So the databases and the tools we are going to use in these modules are we are going to use some basic databases and some databases which was relevant to the plant-based metabolites, and some actual and databases to retrieve the targets, as well as the few softwares to interpret our results, to visualize the network and to do the molecular docking and to do the interaction analysis. So the databases we are going to use here are, so before that I would like to clear the databases are nothing but the data we going to retrieve from the databases. That is some data we are going to fetch from the databases. We call it as database. And certain the data, whatever data we need to analyze we need to analyze via a web link, that we call it as a tool. And when we download and install in the system, we call it as a software. So we have few softwares to be installed for our work. So that will be links will be provided, and I'll just give you a brief introduction about how to install those softwares in your system. Uh, and coming to the databases, I'll just show you the link and I'll just tell you the significance of each databases. So the first and foremost database we are going to use over here was the PubChem database. So which we know very well, it is a freeware of we can check for any kind of chemical substances and its biological activity, that is general physiochemical parameters and its 2D, 3D structure. And in PubChem, apart from those information, you can get a lot more information about the compound other names, specific activities, and 1D NMR. NMR kind of data will also be we get from the PubChem databases, but it is for specific compound. If the compound activity was, compound research data were published in any papers, that data will be linked to this particular page. So we'll get complete information about the compound data from the PubChem. So this was the first database we will be using, where we get the our because our initial input would be the small molecule. So definitely this is the first database. We are going to take the information of the compound, and we get the SMILES format of the compound, and also we get the SDF format. So we use in some tools we use the SDF format, and in some tools we use the SMILES format, that is simplified molecular input linear entry system. So that data we retrieve from the PubChem database. And after PubChem, we'll be screening the compound nature, that is a pharmacokinetic parameters, as our ultimate aim is to create a drug likeness molecule. So we need to do the compound screening. So either we take a single compound or a multiple compound for our research work. It is necessary or mandatory to check for the pharmacokinetic properties. Pharmacokinetic properties are nothing but ADME properties: absorption, distribution, metabolism, and excretion parameter. And also, in addition to the toxicological parameters also need to be verified before when you take any molecule for the research. So those kind of parameters here in our particular module, we are using the Swiss ADME online tool, where we paste the SMILES format which we retrieve from the database PubChem, and we going to check for the that is whether the molecule was water soluble or not, whether the molecule could cross the blood-brain barrier, whether it has whether it has a metabolic activity with the cytochrome enzymes in the liver, and also the toxicological parameters, bioavailability values. These values could be checked with the ADME profiling using the Swiss ADMET server. So it is basically a pharmacokinetic property prediction server. Okay, and so is it clear, participants? Any query in this? So this is a basic thing. Just I'm just for you to know the website links and the details, because at the work, during the work, so it would be easy for you to fetch the data from this PDF. So I'll be sharing this PDF with you. So did I got any query? No, fine. Okay. So coming to the so whatever data so after the compound, the general data we usually going to analyze is the protein. So our ultimate aim is to network pharmacology outcome was the protein. So top 10 protein molecules or top 5 protein molecules we are going to get. So basic details of the protein, that is complete annotation of the protein sequence, structure, and the function details would be present in the that is UniProt database, which is a curated database. Why we use UniProt than NCBI? So NCBI, it contains many duplicate entries for a single organisms. So because if all the publishing data will be accumulated in the NCBI. Whereas in case of UniProt, it was a curated database where if you find any one particular gene, you will get only one particular details for the entire set of gene, and also all the details of the particular protein, that is gene name, subcellular location, post-translational modification, protein-protein interaction details, domain details. All the details will be completely available in the UniProt database. Even the structural data will also be available. So from there itself, we can directly connect to the PDB structure database. So like UniProt, it is a curated database where we can get the protein sequence details as well as the protein structural information will also be available. If the protein structure details were not available, the AlphaFold details nowadays were available in that, which is again AI-based AlphaFold. It's an AI-based server where based on the fold, the structure would be predicted. And coming to the RCSB PGB database, where you can see the protein 3D structure. So as we all we know, the protein structures were predicted using 3 different methods in the wet lab. That is X-ray crystallographic method, NMR method, and cryo-electron microscopic method. So these molecule structure atomic coordinates file would be deposited in the PDB database. We call that as atomic coordinates file. So what atomic coordinates file means the it contains the atoms of the amino acid. So we know as the proteins were made up of amino acids, so each amino acid atom file will be defined in this particular file, and that is the ultimate structure we get. The system will recognize the protein structure in the .pdb format. So here, whatever the protein structure we save, it was with the extension of .pdb. We can edit the protein structure using. We can edit the protein structure using we can edit the protein structure using the text notepad or wordpad format file. We can edit the protein structure, and we can visualize the protein structure using any of the visualization tools like RASMOL, PyMol, or BioVR Discovery Studio. We can visualize using the any particular visualization tools. Okay, so is it clear till now, participants? Any query? Any queries? No queries, fine. So coming to the phytochemical and the natural compound databases. So usually the phytochemical database, we have Dr. Duke's ethnopharmacological database, where if you are aware of your plant source, we can fetch the data of the particular small molecules from the particular plant source. So one plant source may contain multiple molecules, small molecule. So we have each maybe for each database, we'll have a special separate class and how to retrieve, and how that is the hands-on session we are going to perform. So just this is an introductory class where this particular database, it is a database which contain the phytochemical elements based upon its sources, that is leaves, seeds, stem, root. So from different sources, the molecules might be reported in the papers. So those information collectively you can get from this particular database. So this database will contain the plant sources all from the overall world. Whereas we have one more Indian specific database, which we call it as IMPACT, that could also be used. But so in our module, this particular database will be covered. So if possible, we can use IMPACT also, where it is specifically Indian natural plant database, which has complete bioactivity details also, along with the compound structure and all those information will be available in the IMPACT. But this Dr. Duke's phytochemical, based upon the plant part or the plant activity, the compound detail could be collected. And coming to the databases and disease-specific, disease-associated target genes collection from the databases, we use two databases here: therapeutic target databases and the gene card databases. Therapeutic target databases is a highly curated database for the disease-specific targets. Suppose if you want to, this database contains the information about the protein target. So if you put disease name, means you will get the target relevant to the disease, whatever disease you are searching. Suppose it's a liver cancer or a Alzheimer's disease or a Parkinson's disease. If you just put a disease name, it will give some list of targets. So what we do, we copy paste the data from there to Excel format for our research work. So the collection part and all, it will be explained in the hands-on session. But here, this database, it was uniquely for the targets, that is the protein, that is virulent protein or biomarker protein related to disease, which was reported in the clinical trial under different phases by certain drugs. So that was experimental evidence. So we could actually, if we take, for example, if we get around 100 targets for one particular disease from this database, that is very much specific to disease, we'll get the data. And so this is completely based upon the disease name, you will get the data. So this database will help you to get the disease-specific proteins or the marker genes specifically to the disease. And even the gene card, it is also same here. But I could tell in therapeutic, only the clinical trial based data or already research going data you will get. Whereas in gene card, here suppose you get some 100 genes for particular disease. Whereas in gene card, you will get around 20,000 to 25,000 targets also you get. Here in gene cards, it was having multiple relevant data, like curated data will also present from the literature. Many data sets it refer, and it will have all the data collectively. Hence, there will be more duplication. But there you have one more option to refine the data sets based upon the score. We have one probability score over there. So based on the score, we can refine and take the top 200, 300 genes relevant to our disease. So gene card will be helping in to find out the human gene disease. So particularly human genes you will get from the gene card database. Whereas in therapeutic, you can go for organism specific as well as the human genes you will get there. Okay, and coming to the compound. So we are going to collect two sets of data. One is based upon compound-based targets, and another one is disease-based targets. So for disease-based target only, I have told you these two databases we are going to use to collect the data sets. Whereas compound-based target, we use PharmMapper and the Swiss target prediction. We use to collect the compound-based proteins, that is based upon the compound features, pharmacophoric features, hydrogen bond donor, etc., and the molecules. So we'll get the compound data. So that was the ultimate aim of PharmMapper. Whereas in case of Swiss target, it was a probability-based approach. Based upon the compound features, it will go and search the 𝐏𝐥𝐞𝐚𝐬 database, and it will try to fetch the targets. But here the output totally would be 100. But if you take the probability score, you will get, in some cases, you will get around 5 to 10 targets. In some cases, you will get around 30 to 40 to 45 targets. Not much you will get in the Swiss target. But in PharmMapper, based upon the compound pharmacophoric features or the similarity features, we'll get around 300 compounds over there, targets over there. So here also we'll get the target, and in this tool also we'll get the target. Both the target we merge. After that only we are going for the string analysis. So all these server are the databases where we give the input as a, so here we'll give the compound input as the structure, but here we'll give the disease name as the input. Whereas here we'll give the compound, in PharmMapper we'll give the compound SDF format structure, whereas in Swiss Target we use the compound SMILES format. Is it okay? Okay. Then coming to the gene pathway analysis. So whatever the gene data sets we got from above databases, okay. So what we do, we will take those genes and put it in the string database. So string database, it's basically a protein-protein interaction database where it predicts the interaction between the genes. So the interaction could be experimental evidence or a predicted evidence or a text mining evidence or a curated database evidence. So we have different kind of interaction. So you could possibly find between the protein, there would be around 6 lines connected. If 6 lines were connected, then it has experimental, predicted, text mining, all the 6 kind of evidence were there for the particular gene interaction. If it, based on color, we could differentiate what kind of interaction it was. So it was ultimately to find out more the interaction it involves in specific pathway mechanism. So that is why we are going for the string analysis as a first step to interpret our gene network. So after which, whatever data we got from 𝐬𝐿𝐽, we are going to interpret those data with the after the, so whatever data we are analyzing using the string analysis, we do the 𝐏𝐥𝐸 analysis, then we pathway analysis, then we come to the 𝐬𝐿𝐽 analysis, that is gene ontology analysis. As yesterday I have told you certain terms, that is the biological process, molecular function, cellular complex. So those kind of gene, how it was related to these kind of gene ontology term function. So we call it as enrichment analysis. Those analysis we perform with the help of 𝐬𝐿𝐽 analysis. So any doubts, participants, till now? We'll see the main screen. ADMET Lab 2.0. So someone asked, can we use ADMET 2.0 instead of Swiss ADME? Definitely you can use many tools. We have PKCSM tool, we have ADMET 2.0. So we have different tools. So correctly, if you give the input, we can use those tools also, not an issue. And the PKCSM will give more information, but based on the module, so it was a short-term module, so I have given only one tool. So if required, we can go for ADMET 2.0 also. Is it clear? Any other doubts, participants? Any queries?
Ma'am, I wanted to ask, for disease-associated targets,
Yeah.
instead of gene card and TTD, for cancer, can we use CBio portal and Cosmic?
Yeah, wherever you get the target, you can use the portal. That won't be an issue. Our ultimate aim is to, we have many other databases. We have this GenNet, but in certain databases, we have to create a login. Some databases are the paid version. So if you have access to those kind of databases, you can able to check for the target, which was very much relevant to your particular disease. You can take that also. Our ultimate need is to have a list of genes. Even from the literature also, you can go for the genes. Okay, so you're suppose if you are not able to get for the antigenic protein, that is for microorganism, you won't get much in databases. So in that case, you are checking papers and you get around 30 to 40 genes reported for that particular microorganism as a virulent factor. Then we can go for that. No, TTD is not a paid version. It's a free version till now. So if you put a disease name, you will get the data. Why if the compound I'll search be not available in PubChem? So you have to, like, what compound name you have chosen. Like when we go for hands-on session, we definitely check for those information. Like if you have any space kind of issue also, sometimes in PubChem it won't show. Some compound may be patented and that might not be available in PubChem. If you have structure for those compound, then you can give uh, you can so if you have structure for those compound, then we can draw with the help of chem sketch, and we can uh, convert to the specific chemical format we require. Is it okay? And Alexa Shahu, how many databases are required as minimum for a given study? Here you will be using many databases because our basic input was from different databases. I think we use around 8 to 9 databases for the data retrieval. We use PubChem, PDB, UniProt, and we use for disease association we use 2 database, and for compound plant-based compound we use database. So almost we use 10 databases, I think so, for this particular work, because it was collectively, because based on the data only we are going to interpret our results, and even cake database also we use. So next database is a cake database. Hope I have cleared your queries. Shall I move to the presentation? Can I move to the how many?
Hello, ma'am.
Yes.
Actually, I need to ask that if after doing that prediction of it's just a second, ma'am.
Yeah.
Okay.
In data to serve can be
If we select one protein from the databases, that is TDD or gene
Mm,
cards, and then we will do that compound target interaction. In
yeah.
this, we will do this Swiss target prediction. If in that, if we don't get both, I mean, after doing with the compound, we will not get that selected target which
Okay.
we have selected for the docking or further analysis. What should we do then?
If you are like compound based target, it's based upon the features we usually go for it. So if we don't have any idea about the disease based target, then you can go for that. If you're very much specific about your target, and if you have some 40 to 50 targets by your knowledge itself from the literature, directly you can cite that paper and take those compound for the analysis, because all these databases were created from the already existing data sets from the web only. So all these databases, or somehow it was created from the different sources, right? So that is how the databases were framed. So if you're very much specific about the what to say, particular genes to be done, then you can select those 40 to 50 genes, go for string analysis directly. No need to go for all these data sets. Hope I'm clear. Hope you got your answer.
Okay, ma'am. Okay.
Because this
Thank you.
databases, or when you blindly do some work, so we need to so in that case you don't have much idea about the target, so in that case you can go for these kinds of databases. And even this net, when you check now, if you put your disease name, mostly you will get your targets most probably. And so if you got very much relevant target in this gen, where when you go and merge, if you don't get the data set, then you can go for this distant, therapeutic target database alone, that you incorporate in the study. And Indranil, am I clear your query?
Hello, ma'am.
Yes,
Yes,
Indranil.
ma'am.
Ma'am, I have two small doubts. One is what we will use as a positive control and negative control is there in the network pharmacology, and the second is we are using the PDBID with inhibitor bound complex or without inhibitor bound complex. Means hope you are
No,
getting me what I'm saying.
without the complex, without inhibitor only we'll use, because with inhibitor, if you bound na, so it won't bind to the mode particular site what we targeting. So we go for blind docking only. Okay.
Okay,
And
ma'am.
another thing, the databases you asked, like um, So most of the compounds will get from PubChem possibly. So if not, so when you search the name, the alternate names, all those things we need to check in that case. So most of the compound data you will get from PubChem database itself. My knowledge. Maybe maybe zinc database ID, ChemBL ID will also be linked to the PubChem ID. Other names you check means you will have in the PubChem other names you'll have ChemBL and zinc database link also will be there for that. So we'll check for that. So when we do the PubChem analysis, we'll get more details in that. Okay, shall we go for presentation?
Ma'am, my first doubt wasn't clear. Means are we using positive control and negative control for network
So
pharmacology?
here we are not going for any control-based studies because initial input, whatever we are giving, no, for that itself, this 15 days will be not enough actually. So the data sets we collect and we are just going to incorporate those data sets and we are going to analyze those particular data sets to identify the top 10. So if you want very much, you're concerned with very much about the concern. So you can go for the compound already going research. For that also you could collect the data and you can do the positive control study.
Thank you, ma'am.
Simultaneously you can do that if required. Because here our work is completely focused on one compound and getting the data and to make you aware of these databases. So later you can refer papers and you can frame your research work based on the maybe in-depth analysis. If you want to do later, you can contact us to do the in-depth analysis.
Yes, ma'am.
Okay.
Thank you very much.
Yeah, thank you so much. So coming to the cake database, we'll get all the pathway details here. So for that we require the cake database where you get in the string analysis we'll get all the pathways. So to check the related pathways to the disease, suppose if I'm working on particular liver cancer, I need to check for the liver cancer pathway first in the cake and sub pathways to the liver cancer. In same liver cancer, there was multiple pathways were linked. So those kind of information could be checked via the cake pathway analysis. Cake full form, Kyoto Encyclopedia of Genes and Genomes, and all the relevant things. So Parkinson's disease, Alzheimer's disease, all the pathways could be verified over here. So these are the different databases and the tools we are going to use for the next few days. Okay. After which the software, so Cytoscape software, I'm going to use the version 3.7.1, where in this particular version, there was a merge option will be available and we integrate based upon our knowledge. So completely here, I'm not going to use some modules and all to interpret. I'm just going to give you two methods. One, using the hub genes directly to find out the hub gene using the app, Cytoscape app. And another one, knowledge-based approach, where we are going to analyze the pathway from our end. Later, we are going to incorporate in Cytoscape to analyze the network. And coming to molecular docking analysis, we use ACD ChemSketch. If you are not aware, if your compound 3D structure was not available in the database, you can use this particular tool to go for the downloading the software. And you can draw the chemical structure and you can convert that to Smiles format and you can convert that to 3D structure and you can save that in MDL mode format later. You can convert with the help of Open Babel molecular converter. So this software will convert your different formats, MDL to mole form, PDB format, SDF to PDB format, whatever the format we require. This Open Babel software will help us to convert the compound data from one form to another form. And last is the Pyrex. So here we are going to use the Pyrex tool, which was an user-friendly virtual screening software where you can go for multiple compound docking via the Pyrex. So it was actually Autodoc Venus version of Pyrex were available. In that both Autodoc version was also available. Autodoc Vena where we use for virtual screening purpose. And so it was for the interaction between the compound and the target. We used the or molecular docking, that is Pyrex. Last one, BioVR Discovery Studio Visualizer, where we have to download this software to visualize the interaction that was formed via the molecular docking. We will visualize all the type of interaction between the molecule, that is hydrogen bonding interaction, which was formed between the donor and the acceptor molecule, hydrophobic interaction, where it forms via the hydrophobic amino acids, hydrophobic clusters, and also electrostatic interaction in some cases, where it was formed between the charged molecule, and van der Waals interaction, which was loosely bound around the compound. So these are the different things we are going to use for next few days. I'll just tell you how to install each software by pasting the link, and I'll just show you that. The first software we are going to install is Cytoscape. So once you paste your link in your system, you will get this page directly. So you could check here this Windows executive form. So mostly you can download Windows 32-bit or 64-bit based upon your system configuration. So if you just click this download icon, it will be downloaded. So that there is no problem in downloading. And installation also, it was very much user friendly, as it was a Windows-based software. You can install it easily. If you have any queries in that, you can ask in the later class. But just use this link to download this particular software. Any doubt in this? If you're from your end, you are installing, I'll be waiting. Otherwise, you'll be installing later means I'll just go for the next software. You're installing from your hand. If you're not installing, just give a thumbs up symbol so that I'll be going to the next one. You're installing, participants. You're not installing. So I think I'll be sharing the PPT in the drive. Or if you want here also, I'll just share, not an issue. Anyhow, this PDF will be shared at the end of the class, so you can try at that time also. And coming to the next software, 𝐏𝐥𝐞𝐚𝐬𝐞. So it is actually a freeware where you could see here, download 𝐏𝐥𝐞𝐚𝐬𝐞 freeware option. So it will ask us more basic details like first name, last name, your mail ID, your institution and the country, and just give download software. The data will be downloaded. Is it clear? So just click this one and download. It would be downloaded. Yes, any doubts? Just check from your end. If you're not able to download, kindly tell. I'll just share you the zipped folder link. Okay. Participants, kindly check from your end if you can able to download. Otherwise, kindly let me know. Is it okay? Okay. So next software, Open Babel Converter. you have sourceforge.net. So here, this link, if you click means, so if you put this link itself, your software will be downloading. So you could see here, the Open Babel will be downloaded. Okay, so once you install this, the Open Babel, once you install, it will be like this. This icon will be created in your system. And similarly for 𝐏𝐸𝐞𝐚𝐬𝐞, this icon will be created once it was installed. Okay. And coming to the next software, Pyrex. So you have two links for this Pyrex. One is Pyrex sourceforge.net. So you can choose this academic free version, choose option. So all the softwares were downloaded. So you can see here download. And here also it was downloaded. So you could see here the Pyrex software will be downloaded. Okay, so Pyrex, once it was installed, you can see here the app. So like this, it will be, the icon will be created in your system. Okay, and coming to the, is this working? This particular link is not working. If you couldn't download that, you can check this particular link. I have uploaded it in the your drive link, so you can check that. So it was a zipped file, so you can download and install 𝐸𝐸𝐞𝐚𝐬𝐞. Is it okay? So Pyrex you can install like this, and next one, yes, yes, Kaushik.
Sorry, ma'am, it was.
Yeah, fine, fine. That's okay. Okay. So like for docking, we use this, and BioVia, again, it was actually you have to use your mail ID from your college domain to register here. So you can use your mail ID and click the next. It will go for the download option. Go for the download option. Just give agree policy, submit, and it will be downloaded. Okay. So again, this is around 300 to 400 MB downloadable file. Just check for it. So it will be downloaded. So you will be able to download this version, both client and the version. So you just check for this version and you can download the software. Okay, so you just check for this. If you're able to download with that link, you can download. Otherwise, you can give your mail ID and check for this software. You can able to download this thing. So these are the more 5 softwares we are going to use in our work. And apart from that, we have different links. So I'll be posting this presentation at the end of the class. So you can check all the links from your end. And if you have any queries, we can discuss that in detail tomorrow also. This session is completely for the softwares and the tools. I have given brief introduction about all the softwares and the modules. So any queries in this, participants? Any queries? No issues. So I have few thumbs up. Okay. So then what you do by tomorrow, everyone, I'll just share this image, so share it as a PDF file to you in the I have shared this particular session file. So everyone kindly install all the necessary softwares and the tools that we are going to use for the next 15 days, 15 sessions. So is it clear, participants? If you have any other queries, we can discuss the session link for Pyrex. So Pyrex actually already it was there in the PDF document. I have also shared here also. You can check. Okay. So you can install all these softwares and any other queries you want to discuss from yesterday's session, we can discuss that also. Because this session is only for the installation process, so it was very short session, but you want to share here in this. Okay, wait, I'll just share. Cytoscape, I have shared all the links over here. We can do these things in some other compound. Yeah, you have to, your work would be like choosing the compound from your end. You can choose as a group one or a group of compound, or individually also you can do your work and you can combine with your group later also. So, like as Bhavani yesterday, she shared some groups, right? So based on that group also you can go for it. Or you like tomorrow I'll just give you the plant database introduction and I'll tell you how to choose the compound. So that information will be in the tomorrow's class. So I'll give you the database, how to access the database to select the compound. First thing we need to select the compound. Suppose if you're doing research, you'll be aware of compound data. So if you're not doing the research work, then you're not aware of the compound data sets, right? So in that case, I'll be telling you how to choose a compound based upon the activities from the databases available online. So those things will be told tomorrow. Is that clear? Is it, excuse me. Is it clear, Divisha? Yeah. Any other queries from your end, participants? If you have no queries, then we'll wind up the session. So there is no, hope there is no queries. You're all aware of all the tool links and software. So tool links, nothing to explain much detail over here. Every module when we work, so definitely I'll be giving more data about the particular online source or downloadable source tools, and I'll be giving you the working protocol. I mean, I'll be explaining you the working protocol also. Okay. So hope everyone was clear about this today's session. So shall we wind up the session, Mahesh? Then please. I'm not able to access the drive. So you will be having the drive in the classroom link, right? You will be having the drive link in the classroom. Participant, I have no access and not permitted. Um, have you joined the classroom?
Yeah,
Have you
so,
joined the classroom? Mahesh,
so
actually,
everyone, yeah, if anyone have a technical issue, maybe they can contact our events team.
yeah,
Yeah, so I'm,
they don't have access to that if someone is asking, so just ask them to. So you can contact, okay, via the Telegram. So they'll help you, sir, definitely. Fine. So kindly check the yesterday's presentation and today's all the databases and the software links. Try to install all the software so that for the next 15 days we'll work with smoothly with all the softwares and the online tools. So, all the links were provided from in that presentation itself, so that when I work on the specific tools, you can get the link from the particular PDFs. Okay, and general basic information about the tools and the database. So hope everyone clear about today's session. So then we'll wind up the session. Mahesh, you can share the feedback link. Yeah. So feedback links were posted. Kindly fill the feedback link in everyone.
Thank you, ma'am.
Yeah, thank you, participants. Thank you, Mahesh.